Okay, so here we go with chapter 11, section 4, meiosis. This will be the fourth and last video of chapter 11. Okay, so meiosis, it sounds kind of familiar, something like we just studied. Kind of sounds like mitosis. This kind of looks familiar of something that we might have just studied. Well, it is similar to mitosis, but it has some major differences, and that's what we're going to learn about. Okay, so every organism must inherit a single copy of every gene from its parents. And you know this because we've been doing Punnett squares where every offspring inherits an allele from mom and an allele from dad. Okay, can inherit a dominant allele for red hair color from one parent and a recessive allele for hair color from another parent. So you guys should be extremely familiar with knowing that it has to inherit a copy from each parent. Okay, how does it do that? And it does that with something called gametes, and these are formed by a process that separates the two sets of genes so that each gamete ends up with just one set. And that's what we're going to talk about today. We're going to talk about how these are formed and the two gametes that we're going to talk about, and I have mentioned these before. We have egg cells and sperm cells. So these are the two sex cells that we are going to talk about. Okay, so all organisms have different numbers of chromosomes. It depends on what the organism is. This shows right here a Drosophila fruit, which is actually a fruit fly, and it shows that it has 8 chromosomes. Okay. Yep, you can see all 8. Okay, it inherited 4 from the fruit fly's dad, and it also inherited 4 from the mom. So we could go and even label them if we knew exactly where they came from, but I'm just going to okay. And yours are very similar to this, except for humans have 46 chromosomes, of which you have gotten 23 from your mom, and you've gotten 23 from your dad. Okay, these sets of chromosomes are homologous. Okay, so homologous meaning that this is one pair. One came from mom, one came from dad, but they are not identical. They have similar areas where different genes are found. So this could be where the eye color gene is found. So depending on what allele is present, it could be a dominant or recessive from mom or dad. Okay, so these are these are homologous chromosomes. They are matched up here as pairs. Okay, so each of the 4 chromosomes that came from the male parent have a corresponding chromosome that came from the female parent. Okay, and a cell that contains both sets of homologous chromosomes are said to be diploid. So, we'll go back just one more second. So this contains both sets of chromosomes. So this is said to be a diploid cell. Now if you remember, when we've talked before, the prefix di means 2. Okay, so the number of chromosomes in a diploid cell is sometimes represented by the symbol 2n because it's 2 times the minimum number of chromosomes that cell can have. Okay, so for Drosophila, the diploid number is 8 because it had 8 chromosomes, which can be written as 2n. Okay, humans have 46 chromosomes, so humans could be written as 2n equals 46. The gametes of sexually reproducing organisms, so remember gametes are eggs or sperm, contain only a single set of chromosomes and therefore only a single set of genes. These cells are called haploid, and a good way to remember this is they contain half of the chromosomes of all of the other cells in your body. Okay, and this is actually represented by a single N. Okay, so for Drosophila, the haploid number is 4, which can be written as N equals 4, and for humans, N would be, what do you think? If we have our diploid cells have 46, our haploid cells would be N equals 23. Awesome. Okay, so and this is the definition for what we're going to do for meiosis. Okay, so meiosis is a process of reduction division in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. I know it's a big definition, but we're going to go through it step by step so you'll understand it. Okay, it involves 2 divisions. Meiosis 1, and it's followed by meiosis 2. By the end of meiosis 2, the diploid cell that entered meiosis will become 4 haploid cells. So here we go. We're starting with meiosis 1. Now if you remember mitosis, how did we remember the phases of mitosis? PMAT. Well, look. They're exactly the same. They're just followed by a 1 because we are in meiosis 1. Okay, so during interphase, it's similar to interphase of mitosis, except the cell is not getting ready to divide itself. It's actually getting ready to divide itself so that it can reproduce and be used for reproduction. That's the only job of these cells. So, cells undergo DNA replication forming duplicate chromosomes. So remember, we originally all our chromosomes jumbled up. You can kind of see, you can't even see, it's just all jumbled up in there. Then it starts to organize itself with histone proteins, and it starts to organize itself into that classic chromatid shape. Well, it duplicates itself, so we end up with that X shape that we're familiar with for a chromosome. So we have 2, what are these called? Sister chromatids. Okay, so now we are in prophase 1. How can we tell what section of meiosis it's in? We have our 1, means it's in meiosis 1. Okay, so each chromosome pairs with its corresponding homologous chromosome to form what's called a tetrad. Now, let's look at this. So here is our chromosome with 2 sister chromatids. These sister chromatids are identical. Now back here, this yellow guy is the homologous chromosome of the red one. Meaning that it has the same genes, but they might be a different trait on them. So this might have, remember we were saying this might have the trait for red hair color, and over here we might have the trait for brown hair. So this one might come, we're going to say this one came from the dad. The yellow one came from the dad. Red one came from mom. So these came from the parents of this organism. Okay, when they get together, when they all together, they form something called a tetrad. And if you think about the word tetra, means 4. So we have 4 sister chromatids all together forming a tetrad. Okay. And like I just said, 4 chromatids in a tetrad. Okay, so this is where it gets really important of what happens with meiosis. Okay, so when homologous chromosomes form tetrads in meiosis 1, they exchange portions of the chromatids in a process called crossing over. Okay, so what happens is they get together, they end up squeezing together so hard that they end up, pieces break off and be exchanged. So see how they switched legs here? Okay, so this is actually how every offspring is completely different than its parents because of these pieces being broken off. This is how the genetics in sexually reproducing organisms shuffle each other. So this is how they get all mixed up. Okay, and again, this is called crossing over. Okay, this allows for new combinations of alleles. So this is how all organisms look different. Okay, so in metaphase, okay, we have our chromatids lining up along the equatorial plate. Oop. We have our spindle fibers attaching, and they begin to pull apart the homologous pairs. Okay, anaphase is when they really get pulled to the poles. Okay, and then we have telophase, and this is all extremely similar to mitosis. We have telophase and cytokinesis where we form 2 new cells. Okay, so now we have a cell over here, and we have a cell here. Now, remember, we started out with a chromosome with 4 chromosomes. Now, each of the chromosomes that's coming out only has 2. Okay, the 2 cells produced by meiosis 1 have chromosomes and alleles that are different from each other and from the diploid cells that entered meiosis 1. Okay, so now the 2 cells that were just produced enter something called meiosis 2. Okay, unlike meiosis 1, neither cell goes through chromosome replication, so they're just going to stick with what they have. They're not going to replicate those chromosomes that they have. Okay, and remember, our cells now have only 2 chromosomes, or they only have 2 chromosomes. And here we go. Don't have to memorize anything new, because here we go with PMAT again. Only difference, they have 2 after because they are part of meiosis 2. Okay, so here we go, prophase 2 of meiosis 2. Meiosis 1 resulted in 2 haploid daughter cells, each with half the number of chromosomes as the original. So our original member had 4. Now I have half. Okay, so again, During metaphase, they line up, spindle fibers attach. The sister chromatids are now separated, and they're pulled towards the opposite ends of the cell. Now we're in anaphase. Okay, so now after telophase, we now have 4 cells, each with 1 chromosome. Okay, so here let's go over meiosis 1. Remember they crossed over, we ended up with them splitting, so we ended up with half of the genetic material going to each one, and then those 2 sister chromatids split, and we ended up with 1 chromatid of each at the end of meiosis 2. And those 4 cells are now what is used to produce sperm in a male animal. Notice they are all the same size because it's going to be different in females. Okay, in females, only 1 egg cell results from meiosis. The other 3 cells are something called polar bodies. And they're usually not involved in reproduction. It's very rare, and actually what happens is they just dissolve. Now you might be wondering, why is this egg cell so much bigger? Why did it take up all of the stuff from these polar bodies? Well, what happens is that egg, after it's fertilized, needs a lot of nutrients. And there are a lot of nutrients in the cytoplasm. So, in order to have enough nutrients to give the embryo a chance, one egg cell is produced using a lot of the cytoplasm. So it actually takes the cytoplasm from these before they're even produced to concentrate it all in one egg cell. And you can actually see those nutrient-rich cytoplasm if you eat an chicken egg. You can actually see the yolk is actually all those nutrient-rich cytoplasm that is used for the development of the embryo. Okay, so now we're going to compare mitosis and meiosis. Okay, so mitosis results in the production of 2 genetically identical diploid cells. Remember, they looked exactly the same. All it is it's copying the same exact cell. No mutations, no differences. If there are mutations, then there's bad things like tumors, but they are normally genetically identical. However, meiosis produces 4 genetically different haploid cells, meaning they have half of the original chromosomes. Okay, cells produced by mitosis have the same number of chromosomes and alleles as the original cell. So in humans, a cell that has 46 chromosomes goes into mitosis and comes out with 46. It allows the organism to grow and replace its cells. Just like if you got a cut, it allows your body to heal itself by growing new skin cells. Okay, some organisms actually reproduce asexually by mitosis. However, if they reproduce by mitosis, they are all going to be genetically identical. And this is not good. Okay, in meiosis, cells produce by meiosis. They have half the number of chromosomes, so a sex cell that goes in with 46 chromosomes is going to end up with 23 when it comes out of meiosis. Okay, all of these cells are genetically different from each other, and I said that mitosis is not the best thing for organisms to reproduce by, because if everything looks the same and there's no genetic diversity, then if there is a strain of bacteria or a virus it could wipe out the entire population. So, because sexually reproducing organisms are all genetically different from each other, then that means that there is a greater diversity of organisms surviving. Okay, and this is how sexually reproducing organisms produce their gametes. And that's it for my little PowerPoint. I do have a video that I wanted to show you. Those of you who were in class a few weeks ago saw some of this video, but it's really interesting because it actually shows what we've just been talking about. And I wanted to show it to you.
Each and every sperm is one of a kind, carrying a unique genetic package. How is this possible? How can one person produce so many different combinations of genes? The answer lies in the very special way we make sperm and eggs, a process called meiosis. In almost every cell of your body, you have 30,000 or more different genes, spread out on very long strands of DNA, called chromosomes. Most cells have 2 versions of every gene on a total of 46 chromosomes. Exactly half of those, 23, came from your mom, and 23 came from your dad. They come in pairs, where the partners are very similar but not quite the same. The only time they get together is during meiosis. Here's how it works inside a testicle that's making sperm. First, each chromosome makes an exact copy of itself, keeping it attached at one point. They condense, creating an X shape. Now the chromosome partners get together, and the 2, or actually 4, will embrace. They cling so closely, big chunks carrying whole bunches of genes get exchanged between the partners. The cell then divides twice, each time pulling the pairs apart. The final result is a sperm or an egg cell with 23 chromosomes, half the normal number. By itself, the cell is incomplete, but it still holds incredible promise, because every chromosome now carries a combination of genes that has never existed before. All this gene shuffling means that within a single species, there can be an enormous amount of diversity. And the more diversity, the better the odds are that someone will survive to create a new generation.
And that is it. I hope you enjoyed the video today and that you took some good notes. And if you have any questions, we will go over the stuff tomorrow.